Jennifer Fraterrigo is a professor of landscape and ecosystem ecology here at the Department of Natural Resources and Environmental Sciences here in Urbana-Champaign. She is broadly interested in how human activities are changing the structure and functioning of natural and managed ecosystems. The central focus of her research tends to fall into one of two areas: the transport, the cycling of carbon and nutrients, and the maintenance of biodiversity. She approaches these interests from a variety of angles, conducting research at both fine and broad scales, and using a mix of tools including field observations, laboratory techniques, and simulation models. Please welcome her.
Okay, thank you. And it's a great pleasure to be here today and talk to you about some of the lessons that we're learning from research conducted at the Douglas County Farm. So it's great to follow Lowell here, since you already have some introduction now to that study area. And I'm going to be focusing today on really microtopography, so very fine scale patterns for understanding subfield soil phosphorus variability. And I'll touch on phosphorus loss as well in a tile-drained agroecosystem. So I want to start out by acknowledging the research team that has been really important in producing this work. So Lowell, of course, is the one who really helped bring me into this, and then Corey Mitchell has provided a lot of support in terms of helping us collect and run samples. And then two graduate students, one former, one current, Luis Andino, who's now a doctoral student at the University of Waterloo in Canada, and I'll talk specifically about some of the work that he contributed. And then currently Lenarth Ferrari, who's here today and also has a poster. So if you're interested in the results and learning more, I'd encourage you to go and take a look at his poster, which is over there along the wall next to the doors. And then, of course, I want to acknowledge the support we've received to conduct this work from Illinois NREC, as well as the Department of Natural Resources and Environmental Sciences. So very broadly, the goal of my research group is to leverage geospatial data to improve understanding of agroecosystem structure and function, and really support decision-making that promotes sustainability in these systems and resilience over the long term. So I'm going to really focus on just three lessons that I think are pretty critical from what has emerged from all the work that we've done. One is that soil phosphorus is not uniformly distributed in agroecosystems due to microtopography, and this has some important implications. One is that understanding this heterogeneity, this non-uniformity, and specifically locations where we have hotspots of soil phosphorus can really improve fertilization efficiency. And then the second implication or consequence of understanding this heterogeneity is that it can potentially reduce phosphorus loads in tile water. So again, I'm going to really try to come back to these lessons, but also help you understand how we've come to appreciate these in the first place. So as I mentioned, most of this work has been a result of research done on the Douglas County Farm. So this is an 80-hectare, approximately 200-acre farm in Douglas County. So it's located in the Embarra River watershed in east central Illinois. And so this farm, as Lowell pointed out, is part of or embedded within the glacial Lake Douglas, which created this extremely flat landscape and very poorly drained soils. So it's about a 2% slope, and the soils here are classified as Milford silty clay loam. About 40 years ago, there was a parallel drainage system installed at the site, about 4 to 5 feet below the soil surface, with about 100-foot spacing. So 36 of these tile drains are monitored, as mentioned previously, to measure flow, dissolve nitrogen and phosphorus in the tile water, and as you have heard, there have been many studies conducted at this farm. I'm going to talk specifically about work that was done when there was a corn-soybean rotation, about 18 of the tiles split between those two. Tillage was strip till or no till, and then a surface broadcast of granular fertilizer, pea fertilizer as TSP or DAP at a rate of about 92 pounds per acre. Okay, so just a little background. So the figure here shows the arrangement of the tiles, and then the ones that are highlighted in green then are the instrumented tiles. Okay, so I've already said this is a very flat landscape, a slope of about 2%, but there is some variability in that surface terrain as a result of microtopography. And because we have, a lot of data now at our fingertips, we can actually characterize this at a very high spatial resolution. So what I have in the top left corner here is basically a digital terrain map showing that ground elevation. And what it does, highlights these small, subtle, but existing undulations. And we can use these data combined with a few GIS techniques to actually quantify those undulations. So the lower figure shows what happens when we fill those low-lying areas, and then when we subtract these two maps, essentially we get what's in color on the right, which would be a map of depression depths. Okay, so the higher values highlighted in pink are actually deeper depressions, right? So most of this map, as you can see, fairly like what we might consider an upland area, 0 depression, but the purple and pink colors indicate areas where there's actually these very low-lying areas. And I'll mention again, but these LiDAR-based digital terrain models are now completely available for the state. We're actually in the second round of collecting all these data. Not me, but others who collect them and process them and make them available. And this is true for most places in the country now, actually. Okay, so when we do this for the Douglas County farm, we find about 14% of the farm actually has these low-lying areas, or what we'll all refer to as closed depressions here. Okay, so what is so interesting about these depressions? Well, it's probably not necessary for me to tell you, but I will anyway, that these can be the sites of large ponding events. So under extreme rainfall, closed depressions become inundated with water, resulting in these basically ponds or lakes that form out on our agricultural fields. And drain tiles can certainly mitigate the ponding to some extent, but as you can see here from Douglas County, where there is already tile drainage, ponding still occurs. And this could be because the drainage tiles aren't functioning properly, or it could also be as a result of just the capacity being exceeded, and so surface water can't be removed quickly enough. So obviously this can present some very significant challenges for crop production. Ponding can lead to germination failure, as well as the mortality of established crops. And I'll add that there's a lot of projections that this type of activity will intensify in the future. We're seeing more heavy precipitation events, certainly in the spring, so it's likely that ponding such as this will become more common in the future. So we predicted that the closed depressions also contribute to the redistribution of soil P. We know that phosphorus is bound to soil particles, which can be entrained in runoff along slope and elevation gradients, and we typically look at this at much broader scales. But we hypothesized that this process would also be playing out at finer scales, essentially leading to the buildup of phosphorus in these closed depressions. So again, this could be exacerbated by the fact also that there's plant mortality in these areas, so reduced plant phosphorus uptake if crops fail in these areas. And then related to this question of whether or not there was evidence of enrichment of phosphorus in these closed depressions is basically, well, what do we do about this problem? So how do we more accurately characterize the distribution of soil phosphorus when these types of depressions are present? So a little bit, again, about kind of how we typically go about studying the spatial distribution of soil phosphorus. The typical approach, as probably many of you know, is to take a representative field sample based on this gridded type of design. So one composite sample every 2.5 acres, so the equivalent of 1 hectare. in this grid type of arrangement, and this can be intensified at 1.1 acre grid if a higher sampling density is preferred. Okay, so we went ahead and had the commercial sampling done of Douglas County Farm using this gridded-based approach with a 2.2 acre grid, and then we were interested in comparing this to an approach where we actually targeted these closed depressions to collect soil samples from those locations as well and contrast the amount and the spatial distribution of soil phosphorus. So we went ahead and added these additional sampling locations shown here in specifically those areas where we already knew based on the digital terrain model that depressions occurred. So you can see, hopefully, it's kind of small, but the amount of phosphorus, just based on the size of those and the color of those closed symbols, circular symbols, that generally soil phosphorus tended to be elevated in these depressional areas, okay? So, we can basically already provide some evidence to suggest that these closed depressions are, in fact, soil phosphorus hotspots. And again, we can confirm that further if we just look at a frequency distribution of soil phosphorus across all these samples. So this basically shows on the x-axis the concentration of soil phosphorus across different sort of bin classes, and then the percent of samples that fall into those classes. And what you can see is if we look at our 1 hectare or 2.5 acre grid, what we're missing are the high P areas, right? They're just not in the sample because of the regular grid spacing misses, in many cases, those closed depressions. Now, if we look at a more intensive grid-based sampling, so 165 samples, but still with that gridded arrangement, we don't do that much better. We're still missing those high P values, so they're just completely absent from the sample set. And then on the very far end in this panel, what you can see are the distribution for the targeted samples. So that's, again, 165 samples, including both the grid and the depressions, and now all of a sudden we're seeing those hot spots really light up as present in the sample. Again, they're not necessarily frequent. They're a small proportion of the overall number of samples, but they account for, disproportionate amount of phosphorus in the field. All right, further confirming that this is related to depression depth, we can just relate or plot the relationship between depression depth and soil P. And if I just look at the trend relating these variables, including those points that are kind of, I'll talk about in a second, up there on the top. This depth explains about 39% of the total variance in soil P. So those outliers or string of samples or observations in the corner of the plot actually are samples collected from an area that had previously a farm located on it, so it's an old farmstead, and so we believe these are legacy, P legacy samples, and so, in this case, I think those are really important to also consider, but for the purposes of this talk, I'm not going to spend a lot of time on them. And just to mention that when we refit this relationship, excluding those legacy P samples, we see about a 47% R squared. So, 47% of the variation in phosphorus is absolutely accounted for by just the depression depth, suggesting these depressions are key drivers of the spatial distribution of soil P, and that these depressions are also sinks for soil phosphorus. Okay. So that really brings us to the first lesson. I said I'm going to keep coming back to these. And that is just that soil P is not uniformly distributed in agroecosystems as a result of microtopography. And, importantly, depressions appear to be these sinks or hotspots for soil P. Okay, so what do we do with this information? So the next step is to kind of evaluate basically different techniques for estimating the spatial distribution of soil P. So this involves basically looking at different approaches to spatial interpolation. Okay, so I'm going to talk about two approaches that are fairly common, that are out there, and how we've been able to kind of enhance at least one approach using some of the existing spatial data, specifically these digital terrain models that are derived from LiDAR data. So the first method, which is really the basis for the commercial soil P maps that we get from the industry, and that is ordinary kriging. So kriging, in the case of ordinary kriging, it really assumes the distance between samples is the only important process that's creating the pattern in soil P. Okay, so that's kind of our baseline, you might think of. But another method that's available is called universal kriging, and this was really going to assume that there's some trend in the distribution of soil phosphorus for us, and that's due to other spatial processes. And what our data suggest is that it's really this relationship with soil, the depression depth, that can be useful in understanding how or where this trend is and what's driving this trend. So we can essentially introduce this information about depression depth into the model, and this is just an analytical approach, but we can introduce this information about depression depth to basically model the relationship between soil P and its spatial distribution, and then we can interpolate or map that process across space by using essentially the depression depth from the digital terrain model. So we have that continuous surface, and we can then extend it across the entire field. So we did this again for the Douglas County farm. I'm just going to walk you through some of the comparisons that we did, but I'm going to really just focus on the extremes. So we looked again at the ordinary kriging, which we can think of as our sort of commercial method, using that 88 samples from the 1 hectare grid, and then we did sort of all possible combinations with that and the different interpolation method of universal kriging. When you see a T on the end, that's indicating that we're also using now these targeted samples in training our model. And then the NT would be the sample set using that intense grid, but without targeted samples. Okay, but like I said, what I really want to focus on is the two extremes, which would be the ordinary kriging compared to the universal kriging, which again is informed by that information about depth and also uses the targeted samples to train the model. So when we do that, what do we see? The symbols basically indicate, again, a Bray P based on actual empirical samples that we collected in the field. So you can see again what shows up as high would be areas where we tend to have depressions. And then the black and white coloring are the results of the interpolation. So this is essentially our continuous map of soil phosphorus values, indicating how it's distributed spatially across the fields. And hopefully what comes out here is that when we use this universal kriging approach, which again incorporates this covariate of depression depth, we get a much more precise and accurate idea of where those hotspots are. So the high P values would be those that are sort of lighting up as white, relative to the lower P values, more those upland areas, dark colors. So very high spatial accuracy, because these maps are about, I think, 1.5 feet in spatial resolution. So we're really at a pretty fine spatial scale. Okay, so let's see, what does this mean when we sort of summarize all the information across these different sampling approaches and these different interpolations? Well, we get a much different estimate of the total amount of P in metric tons being stored in the same field with these different sampling interpolation methods. Again, our commercial approach with ordinary kriging in the 2.5 acre grid, we can estimate about 571 kilograms per hectare of total phosphorus. And then at the other end of the extreme with the universal kriging and those targeted samples, again, where this is really depth informed and also making use of those samples we collected from closed depressions, we see that there's actually a lot more phosphorus in this field than we otherwise would have estimated. Okay. Again, if we want to see better what is, so that's the aggregate picture, but if we want to see better how it's distributed across space, we can look at these frequency distributions. So same as before, and what you find again is that the commercial approach, all the soil P values are crowded over to the one side of the x-axis, indicating really we're picking up mainly the places where P is fairly low, and we're missing the places where it's high. And that's because, again, we're missing the closed depressions in that case. And then as we move across, these different panels representing, adding more and more information. So if we look at the depth-informed interpolation approach, we do better. We do much better, I think, when we actually have more information to train the model. And we get the best case scenario when we include that information about depth and also include those samples that tell us how much phosphorus is in these closed depressions. All right, so what does this mean for fertilizer? So these are data basically indicating if we look across the field at which pixels or which areas have low phosphorus and would need more fertilizer versus are at a good level and we can just maintain, or areas where it's high and we don't need to fertilize. Again, we see a very different distribution, again, indicating that it can sort of change our practices potentially if we include this information about depth in closed depressions. So most striking about this, I think, would be the bars in dark blue. Again, these represent the most information we can throw at this problem, which is including depth, but also including these samples from closed depressions. And this suggests, in the case of where we have a map using all that information, we actually have a lot more of the area that doesn't need to be fertilized. And that makes sense because, again, we know that some of these closed depressions probably already have a lot of phosphorus in them, and there's no need to be adding more. We do also, though, on the other hand, see that under the low P scenario, places that do need more fertilizer, we do have more area in that category as well under our depth-informed with closed depression map. And so again, we could be potentially missing areas that actually do need more fertilizer if we don't take into account these other factors. All right, so overall, I think what this indicates is that accounting for soil phosphorus heterogeneity, this non-uniformity, can absolutely improve the efficiency of fertilizer practices. It can help us figure out where to put phosphorus, where it's needed most. So avoid the sinks of P, like the closed depressions, and move it to the places that actually do need additional phosphorus that maybe are not being picked up by the commercial approach that is being used today. Okay. So then the last part of this is to talk a little bit about what does this mean for water quality, which I think Lowell did a great job thinking about this in terms of nitrogen, and we've been focusing on the phosphorus side of this story. But again, using the same study location, Douglas County Farm, which, as Lowell mentioned, is well instrumented with these 36 parallel tiles and auto samplers. And so really the question is, how does soil P estimated using these different approaches that I've described relate to tile dissolved reactive phosphorus loads? And I won't say we actually have the answer to that question just yet. We're still working on that. But what I want to do instead is sort of give you an idea, think of what we'll find based on some previous work that we did with Luis Andino. So overall, we expect that accounting for the spatial distribution of these soil P hotspots is going to improve field scale predictions of phosphorus loss. And the reason is these types of results. So this is a study based on a study, again, led by Luis that we published a little while ago. And this was really kind of the basis for a lot of the additional work that we've been doing now, looking at these, the spatial distribution in the soil. And again, this is a project funded by NREC. And What these results suggest is, again, soil test P is very positively and strongly related to dissolved reactive phosphorus yield in tiles. And this is true across the years that we were sampling, so in 2015, 2016, as well as 2017. So this really indicates that the phosphorus stored in the upper soil layer is conveyed to subsurface waters. And, we can look specifically, and we did look specifically at whether there was a relationship between depression depth and the annual yield of DRP in tiles. And, the relationship here is interesting in that we did find a positive relationship, but only under two conditions. One, it was during the non-growing season, and the other, it was in areas that were already below P sufficiency. So really, this is saying a couple things. One, it's saying that where we have excess soil phosphorus, so it's above that threshold of P sufficiency, it's going to contribute to the DRP in the tiles, okay, regardless of the presence or absence or the depth of these depressions, okay? So really the focus needs to be on reducing, the amount of phosphorus in the soils and keeping it out of places, not adding it to places where there's already a lot of it. And then the other thing is that, again, these tiles the closed depressions can sort of add or exacerbate the problem of high soil P through probably a couple different mechanisms that I'm not going to talk too much about right now. Okay. So that really comes to then the third lesson that I want to get across for today, which is that, again, accounting for soil phosphorus heterogeneity can improve the prediction of the P loads in tile water and most likely also help us prevent these losses if we're using it to think more carefully about where to apply phosphorus fertilizer. So with that, I want to end on some management implications. Hopefully, I've demonstrated today that this stratified sampling approach really can add a lot of information, and using this depth-informed interpolation, if we adopt that approach, could really improve the accuracy of these soil phosphorus maps beyond what we have commercially available right now. Kind of related to that then, if we incorporate information from these maps into variable rate P application, then we, I think, have a much better chance of making informed decisions about where to apply fertilizer, specifically keeping it out of closed depressions where phosphorus tends to accumulate and focusing on those areas that actually do need it. And then lastly, I wanted to say a little bit about the water quality aspects of this. So in a lot of closed depressions, these surface inlets are used, kind of shown in the picture there in the top, to very rapidly remove surface water. And these are a serious threat to water quality because they really are connecting that water, which is probably laden with phosphorus, directly to the drain tile, which is sending it out of the, transporting it out of the system. And so a different approach, I think, is needed, and one that maybe makes use of this blind inlet approach, where essentially these blind inlets could be filled with some sort of P-sorbing material to filter particulate and dissolved phosphorus, and preventing it from making its way into drain tiles. And again, specifically within these closed depressions, I think that type of technology could really improve, or has a potential to really improve water quality and reduce phosphorus loss via drain tiles. So with that, I'm going to say thank you and see if we have a little bit of time, I think, for questions.